Assalamu alaikum. Today we're going to be talking about a course which enthuses me. And this is a course on neurological basis of behavior. Have you ever wondered how you are operated upon? How your behavior are organized? How even if you move a finger, what is the mastermind which makes you move this finger? And that is what we are going to be exploring in this course. And that, I tell you, is this cranium which has the mastermind. A universe within this scalp, within this cranium, with thousands and hundreds and trillions of neurons firing away and glial cells supporting those neurons. A universe within a universe. So, what behaviors we exhibit and when? How do we know what to say? What are the words that we learn? Why do we forget? All this is going to be part of this course. Neurological basis of behavior. Neurological basis of behavior has intrigued scientists from the longest period. From the time of Hippocrates, the Muslim scholars, the Europeans, and now in the 21st century. I believe the 21st century is the century of the study of the brain, because the more questions we answer about what happens in the brain, the more questions emerge. What is happening? Are there molecules which decide our future, our present, our past? Are there chemicals which modulate even the highest order behavior, such as thinking or judgment? Are we free to make decisions as we think we are? What do we have in common with the lower order species, the animals who are lower on the phylogenetic scale, the rats, the cats, the monkeys? We as Homo sapiens, what do we have in common with them? Or do we have anything in common? Yes, we do. The brain, the neuroanatomical structures, the neurotransmitters, the molecules, the ionic changes that take place in the brain. So, what we are going to be doing is moving through neuroanatomy, chemistry, biochemistry, physiology, electrophysiology, and this amazing field is based on all these disciplines and the findings of all these disciplines. So, in a neurosciences conference, you see a physicist hobnobbing with a psychologist, and a psychologist hobnobbing with a neurophysicist, a neurochemist, etc. So this is the exciting new field of psychology. It will give you a greater understanding of what psychology is all about, what behavior is all about, and how this cranium and this brain within the cranium and the little brains within the brain make us what we are. Now, the questions that you might have asked yourself are many. The questions is, when did the study of the brain start? And how did it initiate? Perhaps when Hippocrates started thinking about the brain being the center of consciousness and being the center of the body, perhaps that's when. Or we can talk about how does the brain know what to process when and where, and how to link it with what? Is this pre-programmed? Or is there a free will somewhere? Or is there a learning somewhere? Is there a plasticity of the brain? Or is everything rigid and determined? Then we think about, how does the brain grow? Does it grow from the evolutionary perspective, from the smallest amoeba onwards to the higher order animals? Do we share anything? Do we have anything in common with the closest cousins, the primates? Do we have anything in common? Emotions, brain sectors which are modulating emotions in animals? Do we have anything in common? Studies have shown we do, surprisingly. And studies have shown that the Homo sapien cerebral cortex has evolved enormously, and it has given us the superiority that we have. But at the same time, the brain also grows developmentally. The egg is fertilized to the time when the person passes away. The brain is growing, and cells are growing, and the cells are dying. When the cells die, what happens? Neurons do not regenerate. That is interesting. How do we know that? And how does a neuron know where to go? How does a neuron know where to travel? How are the locations determined? Very interesting. What controls our motivated behavior? I feel thirsty. I feel hungry. I feel sad. And the temperature that is regulated within an environment. Why do you, all of us, have the temperature regulated at 98.4? Why does the body, how does the body manage it? The mastermind tells it. And where is this regulation taking place? How do we remember? The smells. You smell a flower, and you remember later on, when you smell another flower which is similar, this is where I smelled this last flower. A rose, a jasmine, a tuberose. All these smells are recorded somewhere. How do we remember the blueness of the sky, the redness of the apple, the shape of the apple? How do I see the apple different from a ball, even though both are round? Now, this is something which is within the brain. How do we know when something has gone wrong? People forget. There are diseases like Alzheimer's, in which there is a degenerative process of the memories. And this takes place within the brain as well. So we, as neurological students of psychology, need to understand what goes wrong as well. We also need to understand where is the pleasure that I feel when I see a friend. The pleasure of having an ice cream, the pleasure of a good game, the pain of losing a friend, the grief of death. Where is it recorded? Are these the neurons which give us this feeling of loss? Or is it, there is, as the poet would say, the heart? And let me say, the heart does not know. It is the brain which decides. So it is within the brain and within the neuroanatomical systems, the questions arise, where in the brain? And what are these little molecules that are running around in the brain, managing us? The molecules of memory, the molecules of emotions, the molecules of anger, the molecules of thirst, hunger, the neurochemicals. And how do we take care of these neurochemicals? Do we modulate them, or do they modulate the brain? What is the electrophysiological changes, the ionic changes that are taking place? The calcium, the potassium, the sodium, the salts, the glucose. If you have 2 bananas more in a day, you have more potassium available to the brain. If you have milk, you go to sleep easily. Milk has tryptophan, and tryptophan is the good molecule, the good neurotransmitter for sleep. So, amazing. Every day, everything that you do, all roads lead to this mastermind, as I say. Furthermore, how do we sleep? And is sleep a behavior? Is what happens when you can't sleep? Things can go wrong, very seriously wrong. And why do we benefit from sleep? Does the brain benefit from sleep? Does the brain ever go to sleep? We don't know. And questions have been asked, and we are going to ask those questions as well. What happens when somebody has a stroke? Or when somebody has an accident? A brain injury, we say, a brain trauma. Very common in Pakistan on the roads. You see children on motorbikes, not wearing helmets. Very easy to hurt your brain. What happens? Because in most cases, the damage is permanent. Is the neuroanatomical location gone forever? Or, as they say, that we only use 10% of our brain. If we use only 10% of the brain, then 90% of the brain is still available for what we call rehabilitation. And how does a person who is recovering from a stroke recover his walking and other motor function? That is something we will be studying here. Why is it, therefore, important for psychologists to understand brain and the functioning of the brain? As one would say, if you need to understand behavior, then you need to understand the underpinnings of behavior. And the underpinnings of behavior come from one and only source, that is the brain. So we are going to be studying the brain. There are many ways of looking at the brain behavior interaction. How does the brain affect behavior? And how is it affected? The changes in the brain affected by behaviors. It is an interaction. So first of all, let me say there are many ways of looking at it. We may look at it, as we call it, in a descriptive manner. Description is observing as is, where is, what is happening, without manipulating anything, looking at things as they naturally occur. Now this gives us an perspective of natural behavior and natural control of the various parts of the brain. Then there is another perspective, and that perspective is what we call the comparative perspective. And I like to say this: look at the animals around you, the dogs, the cats, the donkeys, the horses. You know, we have many animals on the roads and in the homes, even chickens. And you will see a similarity in behaviors. When a dog is angry, it growls. Have you ever seen the picture in which in the movies, when they show an angry man, he is also becoming bigger and he raises his voice in the same manner that a dog would. So there is a comparative perspective of studying the brain as well, because there are similar areas within the brain which coordinate the limbic system, which coordinate the emotional behaviors. Then there is an evolutionary perspective to see how the brain has evolved. Then there is a developmental perspective. The developmental perspective is looking at the brain as it has grown from a single cell to layers of 3 cells, and these 3 different layers of cells, how do they differentiate? And then when they differentiate, how do they become the forebrain, the front part of the brain, the midbrain and the hindbrain? How do they become all this? And how does the spinal cord come in? And how do they connect to the rest of the parts of the body? My fingers and my thumbs, how are they connected to the brain area which controls the movements? And this is through a developmental process. And that is also one prospect of looking at things. Then, what can go wrong? And can anything go wrong? Oh yes, many things can go wrong in the developmental process. And we are going to be studying what happens and what can go wrong and what we need to do to be watching out for what deficits can happen within the brain. We can also look at the continuum of the techniques that have been used, from the naturalistic observation, which we call the ethological or naturalistic scientific observations, to a very controlled laboratory conditions, where even the temperature is regulated, where not just the temperature but also day and night cycles, how many hours the lights would be on and how many hours the lights would be off. So 12 to 12 out of 24 hours. So we can control even the diurnal cycle. We can control the climate within, the temperature within. We can control the feed that we give to the animals. We can control the exposure to the animals. We can control conditions even for humans. So, from the naturalistic, ethological, to the strictly controlled laboratory conditions, there is a dimension in which studies can be done. Let me say, I like to put it this way, that we can study the brain from the molar perspective to the molecular perspective. And when we talk about the molar, we're talking about a holistic and overall, a gross picture of the brain and behavior. And when we're talking about the molecular, we're talking about the smallest single cell that is firing away within the brain and which is nanomolecules, nano particles small, and only a small electrode can record it if you are lucky and if you have the apparatus. And then you can verify it. So this molecular to molar and somewhere in between is what we can study. मोलर इस तरह से है जिस तरह से आप कहेंगे कि एक शहर को आप बाहर से देखते हैं या ऊपर से देखते हैं और शहर के लोगों को दिखते हैं, बाशिंदों को, लेकिन हर बाशिंदे की आपको डिटेल्स नहीं मालूम हो सकती. आपको ये नहीं पता चल सकता कि शख्स ए जो है वो क्या कर रहा है और शख्स बी क्या कर रहा है. शहर की एक तस्वीर आपको नजर आती है, जैसे जहाज पे से आप अगर जा रहे हो तो आप देख सकते हैं. ये है मोलर. मोलिकुलर ये है कि आप बिल्कुल फोकस करके उस शहर के, उस एक इंडिविजुअल के, उस एक घर के इंडिविजुअल को स्टडी करें. तो वो समझ लीजिए कि इस यूनिवर्स को हम न्यूरॉन्स के यूनिवर्स में जब ले जाते हैं तो फिर ब्रेन मोलर और मोलिकुलर स्टडी दोनों हो सकते हैं. इसी तरह से हम ये भी स्टडी करते हैं कि हर इंसान के हार्मोन्स हैं, फीमेल हार्मोन्स अलग हैं, मेल हार्मोन्स अलग हैं, एंड दीज हार्मोन्स रन योर बिहेवियर. इन फैक्ट they run the cycle of your life. These are biological rhythms which run your life. Why do I get up 7 o'clock, 6 o'clock in the morning? एक दिन मेरी आंख सुबह अगर अजानों के वक्त खुलती है तो हर रोज उसी वक्त खुलती है. क्यों? ये किसने फैसला किया? अलार्म क्लॉक ना भी लगाऊं तब भी आंख खुलती है. These are biological rhythms. एक वक्त भूख लगी, खाना खाया, दूसरे दिन, तीसरे दिन, चौथे दिन, हमेशा फिर उसी वक्त, जब तक मैं खुद ना बदल लूं वो भूख उसी वक्त लगती है. सो ये साइकिल्स, बायोरिदम्स, ये हार्मोनली रन है. द साइकिल ऑफ डे एंड नाइट, डायurnal साइकिल, द मंथली साइकिल, द ईयरली साइकिल्स, ये सब साइकिल्स जो हैं ये ब्रेन कोऑर्डिनेट करती है, हार्मोन्स को रिलीज करती है, और रिलीज करने के बाद वो बिहेवियर्स जो हैं फिर वो बिहेवियर्स पैदा होते हैं. अदरवाइज वो नहीं हो सकते. तो मास्टर कंट्रोल अगेन ब्रेन के पास. फिर ख्वाब भी आते हैं. आप और मैं सोते हैं, बड़े ख्वाबों में हम पहाड़ों पे चढ़ जाते हैं, आसमानों पे उड़ते हैं, लेकिन होते बिस्तर पे ही. दिस इज अगेन द मैजिक ऑफ द ब्रेन which makes you go through all this. And there are images within the brain The dreams are manufactured by the brain itself. So we will also be studying. So एक जो मोलर तरीका है ब्रेन को स्टडी करने का ड्रीम्स में, स्लीप में, वो स्कैल्प इलेक्ट्रोड्स लगा के है. लेकिन एक मोलिकुलर तरीका भी है कि इंडिविजुअल इलेक्ट्रोड्स डाल के ब्रेन के इंडिविजुअल एरियाज को स्टडी किया जाए. अब इसमें कुछ हम कैटेगराइज अगर करें तो देखें कुछ सिंपल बिहेवियर्स हैं जो आसानी से स्टडी हो सकते. आसान का मतलब ये कि कदरे आसान. ब्रेन को स्टडी करना मुश्किल है, आसान नहीं है, लेकिन साइंसदान जिस तरह लोग मुश्किल जंगों पे और पहाड़ों पे चढ़ने की एक जदोजहद जारी रखते हैं, इसी तरह साइंसदान लेबोरेटरीज में जदोजहद जारी रखते हैं कि रोज सोचते हैं कि किस तरह से ब्रेन को स्टडी किया जाए और आगे किस तरह स्टडी को ले जाया जाए. तो आसान इस तरह से कि जरा ऑब्वियस है, इट्स मोर कॉन्क्रीट देन एब्स्ट्रैक्ट. मसलन फीडिंग, भूख, प्यास, हंगर, थर्स्ट, रिप्रोडक्टिव बिहेवियर्स जो कि मेजर किए जा सकते. टेम्परेचर रेगुलेशन, जैसे मैंने कहा कि कैसे आपका 98.4 पे रेगुलेटेड रहता है. इसी तरह से इसमें कौन से ब्रेन एरियाज, what are the neuroanatomical locations and what are the neurochemicals which are involved in all these behaviors. Aggression, fear. आप बिल्ली को देखें तो बिल्ली डरती भी है जब कुत्ते को दिखती है, लेकिन उसके साथ उसका एक कभी पोस्चर देखिए कि लड़ने के लिए जब खड़ी होती है तो उसके बाल सर के खड़े हो जाते हैं और दुम खड़ी हो जाती है. ये भी कंट्रोल्ड है. And these are what we call the aggressive behaviors, whether it is the dog, the cat, the monkey, the human. And these can be studied. And then we can also study the hormonal influences in the growth and development, physiological changes, and how are those physiological changes coordinated by the brain or modulated by the brain. लेकिन इसके अलावा दूसरी तरफ ये सिंपलर थे, दूसरी तरफ कॉम्प्लेक्स बिहेवियर्स. कॉम्प्लेक्स बिहेवियर्स वो जिनको स्टडी करना अगरचे तकरीबन 100 साल हो गए हैं लोगों को इसको स्टडी करते, फिर भी ये एनिग्मा है. मसलन लर्निंग, हाउ डू वी लर्न, वेयर इज लर्निंग रिकॉर्डेड एंड हाउ डू वी रिमेम्बर, हाउ डू वी रिट्रीव. ये जो मैं सब बातें कर रही हूँ ये कहीं तो रिकॉर्डेड होंगी, कहीं तो ये मौजूद होंगी, जैसे कंप्यूटर में है. कैसे मैं किसी एक शख्स को देखती हूँ, उसको कहती हूँ ओहो ये मेरे दोस्त से मिलता जुलते, कैसे मेरे मैच किया मेरी आंखों ने और मेरे दिमाग ने, या उनकी आवाज सुनकर मैं कहती हूँ आवाज सुनी हुई है. ये कहाँ जाती है ये सारी इमेजेस और कहाँ से ये रिकॉर्ड होके कंप्यूटर की तरह रिट्रीव होती है ये इंफॉर्मेशन. तो ये प्रोसेसिंग जो है ब्रेन के अंदर जो हो रही है, इस प्रोसेसिंग को भी स्टडी करने की जरूरत है. ये कॉम्प्लेक्स है, इतना आसान नहीं. इसमें मेमोरी है, इसमें लर्निंग है, इसमें भूल जाना, फॉरगेटिंग. जब एक चीज आपने याद कर ली तो भूल कैसे सकते, वो कहाँ चली जाती है. जब मैं कहती हूँ कि कंप्यूटर में सब चीज मौजूद होनी चाहिए तो कहाँ चली गई, कहीं गलत जगह स्टोर हो गई, रिट्रीव नहीं हो सकी. इसी तरह से इससे ज्यादा मुश्किल स्टडी करना. बहुत सारी आपने शायरी पढ़ी सुनी, पोइट्री, जहां पे आप ये देखते हैं कि अटैचमेंट के दोस्त के साथ इतनी अटैचमेंट, इश्क, ये कहाँ है इश्क, दिल तो कहीं नहीं है. तो ये दिमाग में, where is it in the brain that this love and this deep attachment is located? Where is in the brain that addiction of, you name it, we talk about heroin, cocaine, and the different substances, alcohol, people get addicted even to people. So where is this addiction located? Where is the serious psychopathologies such as depression and schizophrenia located? What happens? Is it one neuroanatomical location or many? Is it one neurochemical or many? This has been the most complex. The more complex the behaviors, the more difficult it is to study them. And then even more complex, we have the study of the two hemispheres. If you look at the brain, the cerebral cortex is like this. The two hemispheres joined by what we call a band of fibers, corpus callosum. And this band of fibers joins these two hemispheres, which are independent brains, here and here, and they are joined together. And these cerebral cortices, are they two independent brains or are they one brain? Interesting studies have shown that both of these hemispheres, the left hemisphere and the right hemisphere, have a mind of their own. But then how do they listen to each other? How do they work with each other? One is the language. So when you see me speak, this is the left hemisphere which is speaking. And when you see me making these movements, it is the right hemisphere which is coordinating the space that I see before me. I see there is a chair behind me. I'm sitting on a chair and there's a roof on the top. What gives me that information? It is the right hemisphere and the parietal zone of the right hemisphere which gives me this information. So, as you know, we have gone from the simplest behaviors, eating, drinking, to more complex behaviors like depression, schizophrenia, attachment, and even more complex like the two cerebral cortices, communication, and then even more complex. Where are our judgments? Where is the social conscience that we have? My conscience which tells me, no, this is wrong, don't do this. My mother used to tell me this was wrong. Where is this conscience, the standards, the values, the judgments? That is also one of the most difficult areas to study. Where is our personality located? And that we will find out as we go along, and we will find out that there are complex behaviors that we still have not been able to unravel. We are still on the threshold of the beginning of the study of this enigma, the brain. Now, let me just give you a quick overview of how this science developed, the neuroscience. I feel that this is the most interesting, apart from the computers and apart from the space sciences. I think the spaces within the brain, the universes within the brain, that need to. You have trillions, 10 by 12 billion neurons, and another billion. For each neuron, you have 10 to 12 glial cells. So imagine the population that is existing within this cranium. And that population is what we are talking about. Long time ago, when I was doing my PhD, there was a book which came out written by Wallenstein. And the title of the book was Brain Control. And in that, Wallenstein had very appropriately, that was in the 60s of the last century, had stated that you could control the behavior. This is like science fiction, by controlling the brain. And he gave an example where in a bullfighting arena, he put in electrodes. Delgado put in electrodes in the bull, which was very angry, and there was this audience standing around and waiting for the bull to come in, and the bull just dived at that matador, and Delgado sitting just pressed a button, and the bull just stopped in the middle, looked around as if it was not angry anymore. Delgado pressed another button, and the bull charged again. What a demonstration! So Wallenstein told us then that brain control is not a very far-fetched idea. It is very much there. You know that there are areas of the brain that you can control, and you can use those localized controls to control behavior. Imagine what we can do in the next century perhaps there are behaviors that need to be controlled. Now, the most interesting part of these connections, the brain behavior connection that we've seen, is to solving the mystery as to how does the brain control through interaction of these neurons, control major behaviors. Research is ongoing. Even now as I speak, 24 hours of the day, around the globe, around the laboratories, across, whether in China, whether in Japan, whether in Europe, whether in the US, the laboratories are throbbing with research which is ongoing, which is trying to answer these questions. And these questions are being asked by people working together from the field of physiology, of anatomy, of physics, of chemistry, of biochemistry, you name it, and psychology. In fact, neuroscientists, the largest international organization of neuroscientists, has the largest percentage of psychologists working within them. So we know psychologists are interested and are working. What are the issues within this discipline? The issues that emerge among, we will be going through those issues, but let me say that these issues will come up again and again. And the first issue is localization. Is brain behavior relationship very localized? Is one part of the brain controlling only one behavior? Or is it the total brain which works to control behavior? Some point of view asserts that no, it is one-to-one localization. I will give you an example. If you see my thumb, maps of the thumb have been made of the motor homunculus within the motor cortex, which correspond to my thumb exactly one-to-one. The finger, the next finger, the hand. So, yes, there is localization, but not necessarily so. There are some behaviors which are not as localized. For example, space. The visual space that I have here around me needs several of my senses. My visual sense, my auditory senses, my kinesthetic senses. So I feel, I know. So there is a multiple input, and therefore there is a connection of different neurons which will fire and which will interact to give me this information. Best of us, most of us, would then go by the middle road. We won't say, no, they're not all localized. We won't say, no, they're all connected. We'll say, yes, a little bit of both. And I would like to go by the via media, because that is how nature also works. Nature is not very exclusive on either part. Then the second issue that comes up is the issue of nature versus nurture. So the first issue was localization versus equipotentiality. Localization versus equipotentiality. Whether you're localized or you are equally potential and can do many things. And the second issue that we're going to talk about is nature versus nurture. Is it biology? Is it genetic? Is it determined by fate of the genes? Or can the environment also influence the development of the brain, or what happens within the brain? Now, very interesting, because there are behaviors which are studied like schizophrenia, which have a very strong evidence for a genetic component. And there are behaviors which are precipitated, even though the brain correlates are there, but they are precipitated, or they occur only when appropriate environment is. And for example, these are environmentally influenced, and I'll just give you one very interesting example. When the development of the brain is taking place, there are 3 layers of cells: the ectodermal, the mesodermal, and the endodermal. And these 3 layers are just equal to each other, exactly like each other. Somehow, during this process, development, one cell layer attaches in the middle to another. And that's when we start saying that differentiation will start taking. So one cell which is in the middle will attach to the top layer. So if I move these cells, whichever is in the middle will attach to the whichever is the top layer. So the environment, this layer gets its feelings and its feelers from the cell which is below it. So the environment is important. In the same way, when the brain is growing, the brain grows like this. This is the embryonic brain. The embryonic brain looks like this. So there are 3 humps and then this little tail which is the spinal cord. Now this little hump, there are 3 pieces of this hump. If I cut the middle and put it in the front, this will become the forebrain because it's in the front. Interesting. Whatever is in the front will become the forebrain. So people have done experiments by moving parts of this hump back and forth. So environment becomes important there. The signal comes from the environment. And therefore, even though they are predetermined, the environment makes a difference. So nature versus nurture debate is also ongoing. But again, like we have done before, we will all agree to say that both nature and nurture work together. Nature provides the template on which the nurture or the environment will work on. We refuse to take either side because it's no longer feasible to do so. We have seen that the environment also influences what nature has given, and nature also interacts with what environment provides. So each contributes to the development of growth and behaviors. Then the third issue is, should we control behavior? Should we reduce behavior? We call it reductionism. In order to study why my finger on my hand moves in the way it does, is it necessary to study every single little cell, the molecular cell in the brain which represents my hand or my finger? There's one group of scientists who believes that yes, it is important to reduce behavior to its simplest possible component in order to be able to answer questions. On the other hand, there are others who believe that if you break down components, you will never be able to see behavior as a whole. So if you want to see my hands moving in the manner they do, you have to study the whole musculature and the nerves which control this, instead of studying one single nerve and one single neuron atom. So again, this is the third controversy, whether to go and reduce behavior to its simplest molecular micro components, or to study behavior in a molar fashion or in a naturalistic fashion as it occurs. Behavioral neuroscientists again have used both procedures. They use both the molecular and the molar, and then they put it together to get a clearer picture. So there are techniques which will break down behavior in its components, and there are techniques which will see, and then we can superimpose. It's like a camera picture which can then give us a multi-dimensional picture of what is happening. Remember, behavior is multi-dimensional. Brain is many more dimensions. So in order to study this, we have to study both aspects and put it together. Then there is another, which I'll go to in later, the issue. Do we need to take all behaviors to the laboratory? Or do we need to study in the natural environment as well? There is a group which believes, yes, you have to control all variables. You have to control the level of neurochemicals. You have to control the ions that are released in order to be able to effectively predict and to study what is going on within the smallest synapse within the neurons, the communication within the neurons. Okay, but then there is a group who believes that, of course, it's important to study neurons at the smallest level, in the laboratory, in controlling, but then that is a controlled phenomena. That is not how behavior naturally occurs. There is a behavior which is a free roaming behavior, and we needed to study normal, naturally occurring behaviors if we have to generalize to normally occurring behavior. Again, there is a compromise. People do both and put it together, and these conferences of neuroscientists, this is where people are talking about all as well. We've talked about various perspectives. Let's talk about the different beginning of the roots of neurosciences. As I said earlier, the roots of neurosciences are, if you trace it back to prehistoric times, perhaps there are many. But let me start with the father of medicine, Hippocrates. He stated, and this is on record, that the brain was the seat of all sensation, thought, emotions, and even judgments. So, perhaps we can start a study of the brain, the roots of the study of the brain, emerging from Hippocrates' statement. Very interestingly, I have a large number of contributors from the Renaissance and the European tradition and the Russians but there is one which I found only once in a book, and I have tried to trace it again and again. I have not been able to do so. There is, when you study the brain is covered by a thick layer, a very thick layer of membrane, which is called the dura mater. D-U-R-A-M-A-T-E-R. Dura means durable or thick, and mater means mother. Now, this doesn't make sense. A thick mother covering the brain. And then there is another layer within this, which is known as the pia mater. P-I-A. Mater is M-A-T-E-R again, the small mother. How did these two names come about? I tried to locate this reference and I have not been able to locate it, but I'm going to try it again. And maybe in a later class I'll be able to tell you. This reference is to the Arab Muslim scholars. They had studied the brain and they had dissected the brain and they had seen this thick and a very protective, strong protective layer which they call the means mother, cover, so And the when the Renaissance, during the Renaissance, the Latins translated exactly in the concept of mater, which is mother. Very interesting. So this contribution, early contribution in the brain sciences were also due to the Muslim scholars, the Arab scholars, who had dissected the brain and who had identified the different parts of the brain and related the different meninges that we see, the coverings, the meninges, the functions of these meninges, the cerebrospinal fluid. So we will be talking about that later. Then we have the Renaissance, in which we see names from the European traditions emerging. Franz Gall was the first one who talked about the holistic, not looking at individual parts of the brain, but the whole brain itself. And the specialization that he proposed was called phrenology, studying of the bumps and the shape of the brain, the scalp, to identify the characteristics of the individual. Then we had Florence, who tried ablating. Ablating means, ablation का मतलब होता है जैसे ये ब्रेन है तो इसका को काट दिया जाए, कुछ हिस्सा निकाल दिया जाए, सर्जरी कर दी जाए. तो Florence in the 1800s, mid 1800s, tried doing surgery on the pigeon brains and the chicken brains. And he demonstrated for the first time that if you damage one particular area, you damage the functions. So, पहली दफा उसने ये demonstrate किया कि brain damage के साथ functional damage होता है और biological damage यानी body में damage भी होता है. उसके बाद बड़े interesting European discoveries होनी शुरू हुई. एक जो बहुत ही interesting है और जिसका impact अब बहुत ज़्यादा हुआ, लेकिन जब पहली दफा उसने अपना पेपर पढ़ा एक कॉन्फ्रेंस में, तो सुनने वालों ने सुना और दरवाजे से बाहर निकल गए कि ये क्या कह रहा है. ब्रोका. And one small story is which I read from Paul Sagan's book on Broca's brain. The title of the book is Broca's brain. वो कहते हैं पेरिस में मैं एक जगह गया तो एक गली में एक बड़ा धुंधला सा मकान था जिसमें धुंधली सी म्यूजियम थी जिसमें बहुत मट्टी पड़ी हुई थी और सेंटर में एक पेडेस्टल पे एक वाज पड़ा हुआ था जिसमें फॉर्मलिन के अंदर एक ब्रेन पड़ा हुआ था, उसके नीचे लिखा हुआ था ब्रोका's brain. Broca is a Frenchman who first discovered, I'm articulating, you can see, I'm speaking. Broca is the first one who found which particular area in the brain is responsible for me articulating, speaking. So, this was in the mid 1800s, and Broca's area in the brain is now known as the speech articulation area. Then we have many other names. Wernicke was another one who came up with the speech, speaking. One is articulating and one is understanding, comprehending and receiving speech. So, if I listen to you and you say something to me and then I respond to you, say, what is your name? I say, my name is such and such. So, there are two processes involved, receiving information and articulating. Now, this receptive speech was Wernicke's area and they're still in the brain known as the Wernicke's area and the Broca's area, and they coordinate and work together. So, we also see a large number of, and I'm going to go into the sequence of these events. But let me just quickly give you who else is involved. Physics. There is a base of neurosciences in physics. And one of the earliest studies in psychology, if you are a student of psychology and you have studied psychophysics, you will know that psychophysics is a branch of psychology which assesses and measures the functions of the physiological and the physical responses. And these are measured in physical terms. So, physiological and physical responses are to a stimulus, and this stimulus-response relationship is then established. Then we have other names, but we will be talking about them later. Suffice it to remember that neurosciences is an exciting discipline which has grown out of multiple disciplines. This is still growing and still has multiple inputs from many disciplines. It has its roots, and understandably so, in physics, in chemistry, in ethology, in Darwinian evolution, in sociology, in sociobiology, in psychology, and in various branches of psychology, in ethology. So, you name it, and each in chemistry. So each one of these branches of sciences has contributed to and is contributed again by the behavioral neurosciences or the neurological basis of behavior. Because brain is a very complex phenomena in itself, a universe in itself. Therefore, in order to study it, we'll have to study the beginnings of this discipline. We have to go across the globe to see how it developed. There are traditions in Russia, there are traditions in Europe, in Germany, in France, in England, in Spain, in Italy, and then this was taken by the US, and majority of the Nobel Prize winners now have been working in the US, and we'll be talking about this in detail a little later. So we have now to go into the details of the names that stand out in the contribution of neurosciences. The father of neurosciences, biopsychology, neurological basis of psychology, neurological basis of behavior, is known to be Lashley. The father of relating physiology to behavior is Pavlov, coming from Russia. And the father of neuropsychology is Luria, again coming from Russia. The father of neurophysiology, or study of electrophysiological connections, is Sherrington, coming from England. So we have these names: psychophysics, Weber, Fechner. And then we have Sherrington, Lashley, William James, Helmholtz, Young. We also have Pavlov, and these names coming up from the European traditions. One of the most important contributions of studying the neuron has been to identify the location of the neuron and to see how the neuron, the body, the soma of the neuron looks like. And that is the neurons, the picture, and we'll talk about it in the next